Hi class, today we're going to be starting chapter 9, Fundamentals of Genetics, so kind of our background in genetics. We're going to start with Mendel and his experiments. He is considered the father of genetics, Gregor Mendel. And so if you're using your notes packet, we're basically just going to be going through what his experiments were in this first section. So to start with, we have a definition of genetics. It is the field of biology that's dedicated to understanding how the characteristics are transmitted from parent to offspring. So the key part here is that it's the field of biology that looks at how things are transmitted from parent to offspring. And the father of genetics is Gregor Mendel, and then heredity is the actual transmission of characteristics. So we're looking at the actual passing of the characteristics from parents to offspring in the term heredity. So Gregor Mendel, he is the father of genetics. He's the one that came up with the ideas that we know now to be dominant and recessive traits. And he really started this whole process of being able to look at why we have different traits for certain characteristics or different possibilities that can show up in an offspring. So he was an Austrian monk. He was a science teacher in the mid-1800s. He worked with pea plants in his garden, and he decided to study 7 kind of main characteristics of peas. Some of them that we'll look at today that are real distinct or real obvious: purple flowers versus white flowers, tall plants versus short plants, flowers at the end of the plant or flowers in the middle of the plant stem, round seeds or wrinkled seeds, or green peas or yellow peas. So they were pretty simple to see. They weren't things that he had to have a microscope to be able to do anything with. It was pretty much just characteristics that he could see. And he wondered why they were different. So these are some of the characteristics. Each characteristic had 2 different traits. For example, tall versus short, purple versus white. You can see here some of the other characteristics he looked at: seed shape, spherical or round and wrinkled; seed color, yellow and green; pod shape, inflated or constricted; pod color, green or yellow; and then tall or short and where the flower was. Again, if you want to look at these in your book, page 176 has a chart you can look at or refer back to. So Mendel's experiments, we're going to go through the steps here. He started off with 2 pure plant groups for a particular characteristic. So as an example, let's say he looked at purple flower and white flower. So he picked a characteristic and went with that. He called this group the parent generation. That's what that P stands for. And he made sure that they were pure for that trait. Now, in pea plants, they actually self-pollinate, meaning that they don't cross their genetics or their traits with any other plant. So since they self-pollinate, he was sure that they were pure for that particular trait. They didn't have anything mixed in with their trait. Step 2, he cross-pollinated these plants, and this is kind of a key part here at step 2. So he allowed the purple plant to pollinate the white plant. So as our example here, purple flower plant pollinated the white flower plant. And when he finished that generation, or when it did actually reproduce, he called it the F1 generation, which stands for first filial generation. Now, in order to cross-pollinate, he actually did this. In this picture, you can kind of see what would happen. He took the purple plant and he took a paintbrush and got some pollen from that purple plant and transferred that pollen to the white flowered plant. And then once they matured into a pod and he planted those, he would see his first generation offspring. We'll be going through those results in just a few minutes. Okay, step 3, when these seeds matured from the F1 generation, he counted and recorded the traits of that offspring. So basically he counted purple, versus white to see what he could find. He actually found in this example that all the plants were purple. Now we're going to put that in just a few minutes somewhere else, so you don't have to write that in just yet. Step 4, he then allowed the F1 generation to self-pollinate. F1 generation self-pollinated, and that just means that it's doing what it normally does. He didn't transfer any pollen to another plant, and that produced the next generation, which was the second filial generation, or F2. Okay, so we're going to make some Punnett squares, and in your notes packet, you can take a look at the bottom of the notes packet to be able to fill these in. So in your notes packet, it actually says F1 Punnett square, but we're going to change that to P Punnett square. So in your notes packet, if you are following along there, make sure you change the F1 to a P. And we're going to look at the purple versus white. So you may or may not have done Punnett squares before. This is kind of a little introduction. We'll be doing these more in the next few days. Purple flower is going to be a big P, and a lowercase p is going to be white flower in our example here. So you're going to write that off to the side somewhere. Now personally, when I make a lowercase letter in these monohybrid crosses or any type of Punnett square, I put the line over top of my lowercase letter because so many of these letters look very similar when you write them, whether they're lowercase or uppercase. So if you put that line over top of the lowercase letter, it just helps to clarify that that's what you're doing. So in the Punnett square, the parent generation was a purple plant that was purebred for its trait of being purple, crossed with a white plant that was purebred for the fact of being white. And when we make that Punnett square, we take parent 1's information and we put it across the top, and we take parent 2's information and we put it down the side. We then pull in the letters into each box, so big P, big P, little p, and big P, little p. So all of these offspring show a big letter. Now in this case, it's a normal type of dominant trait, so big P represents purple. So all of the offspring, or you can put a percentage, 100% are purple flowers. Okay, so that's what he found in his parent generation. So now we're going to do the F1 Punnett square. And in your notes packet, if you're following along there in the notes packet, it says F2, so please change that to F1. He then allowed this plant here to self-pollinate. So it's going to end up being big P, little p, crossed with big P, little p. So parent 1's information across the top, parent 2's information down the side, and then we're going to pull the letters in just as we did before. So this time we have a unique one. We have one that shows 2 recessive letters. That individual is going to show white flower. So we have 3 here, individuals showing purple. So 75% are purple and 25% show white. In the Punnett square, you should be able to see that as the results. So in this generation, he was able to see that now, instead of just all showing purple, he was able to see that that white trait showed back up. It skipped a generation, if you want to say that. And so since it skipped a generation, he realized that the big P in this case was dominant, and the recessive, or one that was opposite of the dominant trait, was showing up every other generation in this case. So if you flip the page over in your notes packet, there's a place to put our results. In the F1 generation, something that you can abbreviate this down to, they all showed purple. Go ahead and put that in the square that says Mendel's F1 results. And then this goes in that space as well. He named this the dominant factor. In Mendel's F2 results, he had 75%, 25% between the two traits. And he named the second trait recessive. Okay, and then Mendel came up with some laws as well. And so our two laws are the law of segregation, which if we think about what that word means, segregate, if we segregate something, we separate it out from something else. And that's really what's happening. It happens in meiosis. He said that a pair of factors is segregated or separated during the formation of gametes. So if you see here, we have our factors. In this case, they're Ys. They get separated in this process of meiosis so that we have one trait in each egg or sperm cell. And so what does this mean? Each egg and sperm receive one factor from each parent. And again, this occurs during meiosis. So basically, all the traits are separated into the 4 different cells that are being produced in meiosis. The law of independent assortment, if you think about what that word means, independent is usually being by yourself or solo. Assortment means you're sorting things, so you're independently separated from each other. And this definition is factors of different characteristics are distributed to gametes independently. So what does this mean? The factors for different characteristics, like blonde hair and blue eyes, those are 2 different characteristics, are not connected. We don't see traits being passed on together. They're not linked. They're not stuck together. Instead, they're all independent of each other, and they can all be passed independently of each other to the egg and sperm cells, which then would link up and you'd get individuals that don't necessarily have traits that have been linked together from their parents.